Good day and welcome everyone to a full-scale application of phytoremediation to address 1,4-dioxane plume. Conference call hosted by Scott Hartshaw. My name is Corinne and I'm your event operator. During the presentation your line will remain on listen only. If you require assistance at any time, please key *0 on your telephone and a coordinator will be happy to assist you. I'd like to advise all parties the conference is being recorded for replay purposes, and now I'd like to hand over to Scott. Please go ahead.
Thank you, Corinne. Appreciate everybody being here and taking the time to join us for this presentation. I think we've got a great one today. It combines sustainability as a core value of Parsons along with remediation of an emerging contaminant, the 1,4-dioxane plume. So hope you enjoy what we're about to see. Tracy? Our standard housekeeping items, as you heard, our phones are going to be muted. You can still submit questions using the Q&A feature within WebEx, and we will read and answer those at the end of the call. If you do choose to submit a question, we'd ask that you would give us your name and location in case we need to follow up and give you additional information after the call. We will have an opportunity to share more direct information. One other thing I would mention is that this webinar has been approved for continuing education. So if you are in a position where you need the continuing ed PDH hours, just send me an email and I will get you the sign-in sheet and we will get a certificate out to you right away. A brief agenda. We're going to have some introductions into the speakers that we have. As we start all of these, we will begin with a core value moment, and then we will have presentation with time saved for some Q&A as time permits at the end of the presentation. Our presenters, Tracy Obie is a Parsons project manager in the Charlotte office. She brings to us 18 years of experience in environmental studies, remediation, and emerging contaminant investigation. Amber Brenzikofer is a senior project manager with 30 years of experience in the area, also dealing with remediation and phytoremediation and construction management. And with that, I will turn it over to Tracy.
Okay, thank you very much, Scott. I appreciate everybody being here today to listen to our talk. At Parsons, we have our core values, and obviously safety is one of our most important ones. So we like to start off these calls with a core value moment. And we've selected a safety one for this month because October is actually National Fire Prevention Month. And it's so important to take the time out at this time of year before you head into the holidays to think about your fire prevention plan with your family, talk about fire safety, test and change out your fire alarm batteries. Make sure you've got a fire extinguisher and that it's up to date, not expired, and that your family members know how to use it and when to use it versus when to, escape from the fire. 3 out of 5 home fire deaths resulted from no working smoke alarms. So make sure that you're regularly changing your batteries. I know some are hardwired into your electrical system, and those usually have to be changed. They generally only last about 10 years or so, so make sure that you check those regularly and change the batteries. I think we all know the annoying chirps that you can hear, but it's a good reminder to get those batteries changed. The other thing to keep in mind with this is carbon monoxide is the #1 cause of accidental death, and many people don't have a carbon monoxide detector in their homes. So it's important to think about doing that as well, getting one. I know some people even travel with the travel one because there have been some issues with hotel rooms. So just make sure that you take some time this month to talk about that with your family and make a good plan. Have an escape plan that everybody is aware of. That way you won't have any issues. If there is an emergency, you'll know exactly what to do and everybody will know exactly where to go and how to handle it. So with that, I'm going to move on and go into our presentation. So we're talking about a 1,4-dioxane plume that is at a site we have in coastal South Carolina. It is a site that formerly housed a pack parts cleaning facility for a nearby operations plant down the road. Operation of the plant of the activities there were discontinued in July of '91, and we've been doing ongoing groundwater sampling investigations since the 80s. 1,4-dioxane and some other contaminants, low-level chlorinated solvents, are present in the shallow groundwater. And historical source areas appear to be a former septic tank or a leach field with a wastewater collection sump that was immediately behind the facility and had some leaks. There is currently a deed restriction prohibiting groundwater use on the property. The front part of the property was divested to another owner, so we only have a small parcel left in the back, which is where the environmental liability and the majority of the plume is located. So the only shallow groundwater is contaminated, and there is no use of that for potable purposes. A little bit about the site geology, hydrogeology, just to kind of give you an idea of what we're working with. We monitor mostly the upper aquifer, which is it's very shallow. It's about 2 to 10 feet below ground surface, and it ranges from about 2 to 14 feet in thickness across the site. And underneath that is a very tight aquiclude. It's a marl clay that's about 80 to 100 feet thick. And all of the drinking water wells would be installed below that. So we're only talking about superficial groundwater that we're looking at here. And that's where all of our monitoring wells are located. We've already completed some remediation activities at the site. As I said, the site has not been in operation since the early 90s, so we did a dismantle and removal. All the buildings on site were taken down to the slabs, which do remain. So about 18-inch thick concrete slabs were left remaining. We've been doing long-term groundwater monitoring at the site since 2001. Again, 1,4-dioxane being the main COC, much higher than anything else in the groundwater. We did do some underslab sampling in 2004. It did not show any major hits in the soil under the remaining slabs. And we did a lower aquifer investigation below that marl in 2018. Again, not anything that we needed to be concerned about with that. So it is focusing completely on shallow groundwater. And this picture shows some groundwater 1,4-dioxane isocontours. So you can kind of see we have the majority is in the main, coming right off the back of the slabs. This is the slab area where we have slabs remaining, and these are the wells that are the most highly contaminated. This blue line is showing approximate groundwater flow. We've also done some other remedial activities that include the installation of a phytoremediation system that was installed in 2007. We've sampled as well, and Amber's going to talk a little bit more about that in a minute. And we also did go back and remove that slabs, the remaining concrete slabs, and the underlying soil. We just completed that work, so we'll talk about that some more as well as we go along. At this point, I'm going to turn it over to Amber to talk about phytoremediation and the installation of the system at the site. Amber?
Okay, thank you, Tracy. I want to briefly describe phytoremediation for those of you that may not be as familiar with this technology. So phytoremediation is a set of processes that uses plants to destroy or remove contamination in groundwater and soil. Plants use several mechanisms in phytoremediation, including phytoextraction, which is the uptake and translocation of contaminants in groundwater and soil from roots to above ground portions of plants. These plants may grow for several months and then are harvested. The plants can be landfilled, incinerated, or composted depending on the contaminants and contaminant levels. Typical crop species are used, and those can be easily harvested and disposed of. And you typically see this in metal contamination in the soils. Phytostabilization is the use of plants to immobilize contaminants at the interface of the roots in the soils. This is also used mainly for heavy metals in soils. Phytodegradation is the breakdown of contaminants taken up by plants through compounds such as enzymes that are produced by the plants. The pollutants are degraded, used as nutrients, and incorporated into plant tissues, or the byproducts are volatilized out the leaves. A good example of this includes hybrid poplars that have enzymes that break down TC within the plant to CO2 and chloride. Phytostimulation is the breakdown of contaminants in the soil through microbial activity in the root zone that is enhanced by the presence of microorganisms such as bacteria that consume or degrade the organic substances. Natural substances released by the plant roots, such as sugar, alcohols, and amino acids, contain organic carbon that act as nutrient sources for the microorganisms. Plants also transport atmospheric oxygen to the root zone. In phytovolatilization, contaminants are taken up by the plants and transpired through the leaves to the atmosphere. Many organic compounds that are transpired by a plant are then subject to photodegradation in the sunlight. A combination of phytoremediation processes may occur at a site depending on different contaminants or different exposure concentrations. Hydraulic control is the use of plants to influence the movement of groundwater through the uptake and consumption of large volumes of water. From an EPA technical fact sheet on 1,4-dioxane from 2017, phytoremediation through the use of plants such as poplars, willows, and cottonwoods have been shown to be effective to remediate 1,4-dioxane in soils and groundwater. Next slide. So as Tracy mentioned, in 2007, we planted 42 white willow trees on site in 3 zones on 8-foot centers as part of a pilot project. The tree cuttings were 8 foot long and about a 1/2-inch caliper, essentially long sticks. And I have a photo on the next slide that shows these. Boreholes were drilled to approximately 3 feet below ground surface at each planting location using an auger drilling rig. The holes were backfilled with a porous rooting medium and fertilizer pellets. In 2011, the trees ranged in size from 18 to 45 feet tall with an average tree height of 33 feet. The next slide has photos of the initial 2007 tree installation showing the 8-foot cuttings. Tracy's in the bottom left-hand photo for reference. The next slide. Here are the white willow trees in 2011 when Tracy and I did tree measurements and evaluated the overall health of the trees. And this is typical growth for a white willow after 3 years. The tree root depths were also investigated by digging soil cores by hand to determine if there was any interaction of the roots with the groundwater. We did several soil borings near the trees where we encountered tree roots down to 4.5 feet below ground surface, where the groundwater table was at 4.4 feet below ground surface. So the roots had reached the water table within the 3 years. Next slide. So in addition, Tracy and I collected tree tissue samples from 9 trees that included tree cores and leaf samples, and they were analyzed for 1,4-dioxane and other site-specific VOCs. The samples were analyzed by Utah State University's Water Research Lab, who specializes in phytoremediation research and tissue analysis. The highest 1,4-dioxane concentrations occurred in the tree core samples in the 2 westernmost tree stands. Concentrations range from 46 to 148 parts per billion. There was less concentration in the leaf samples, indicating that contaminants had potentially volatilized out of the leaves. Next slide. In 2012, we visited the site again and took tree measurements that showed that trees had increased an average of 1 inch in diameter from the previous year. We also investigated if native trees downstream of the site were taking up contaminants observed in the down gradient wells. 16 tree core samples were collected from existing willow trees and downstream native trees and analyzed for 1,4-dioxane by the Utah State Water Research Lab. We updated the conceptual site model to include calculations performed to assess the potential migration of 1,4-dioxane under current conditions. Next slide. So 7 tree core samples, shown here as yellow dots on the map, were collected from existing willow trees, and 9 downstream native trees were also analyzed. The results showed that the 2 downstream native trees, you can see the 26 and 37 parts per billion concentrations above the groundwater flume, and Tracy just drew a circle around it. The rest of the downstream trees that we sampled had non-detect concentrations. On site, the tissue concentrations ranged from 11 to 77 parts per billion. And as you can see from the contours, the 1,4-dioxane concentrations range from 660 to 19,000 parts per billion below the on-site trees. To quantify the amount of 1,4-dioxane mass removal from the site by the trees, you can estimate the tree canopy area and using known evapotranspiration rates for white willows. The plant stand was estimated to be taking up about 250,000 gallons of water per year. Using a basic formula that included a transpiration stream concentration factor for 1,4-dioxane, the groundwater concentrations near the trees and the total water transpired by the plant stand in a year, the total removal rate of 1,4-dioxane was estimated at 9.3 kilograms per year. That number was anticipated to increase as the trees got older and consumed more groundwater. I now want to turn the presentation back over to Tracy, who's going to talk about groundwater modeling.
Okay, thank you, Amber. Yes, part of that investigation that we did in 2012 was to look at overall 1,4-dioxane concentrations and do updated groundwater models and update the site conceptual model as well. So we started off by looking at 1,4-dioxane concentrations over time, and I think it might be a little more difficult to see on this slide, but there's a line right here that shows where the year when we installed the phytoremediation system. So you can see it goes right about here, and that's showing that we did not have any significant change in groundwater concentration after the system was installed. So from 2007 to 2012, concentrations were generally within historical ranges. There was even a little bit of a spike. So what that told us, and that showed actually even with the modeling that just for dispersion purposes and natural degradation, we would have expected the concentration of 1,4-dioxane in the major down gradient wells to have decreased more than that. So we determined at that point that there probably was an additional area of a mass somewhere feeding additional 1,4-dioxane into the groundwater. And we thought back to the slab samples that we had done. As I said, there was a whole large area on site of underground of remaining slabs that had been the base of the buildings that were on site that had not been removed. And we figured that it was likely that there was additional concentrations under there. So what we did was we decided to remove those slabs and take a look and also remove any potentially impacted soil that was under there. As I said, we did collect under slab samples in 2004, but we just went and hit a few spots around the slab with a geoprobe and collected samples right there. So if there were any pockets or pools, puddles, or so to speak, of remaining material, we would not have been able to identify exactly where that was. So we went out this year. We actually went out just last month, and we did our first step in our remediation path forward, and that was slab and soil removal. The objectives of that were to get rid of any remaining residual source material that might have been under the slabs. And we started off, we took out about 1,500 square yards of on-site building foundations. We also removed all the vadose zone soils underneath those. So originally we were going to use a PID to check for potential areas of contamination and only remove either visually or PID impacted soils. But the groundwater was very shallow. It was only about 3 feet below the slab. So we ended up removing most of the soil from underneath where we had taken the slabs out. We took sidewall samples. We had originally intended to take bottom samples as well, but because we dug all the way to the groundwater table, there wasn't any point in taking bottom samples. So the sidewall samples, we dug up as much as we could. When we took the sidewall samples, we got back non-detects all around for 1,4-dioxane, so we were successful at removing whatever might have been there. But we also discovered an area where that former sump had been what we originally thought was the historical source area, and I'll show you a picture of that. They did find a sump vault, a concrete sump vault, and around that sump vault was some sludgy-looking materials that had high PID hits. So we dug all of that up. So hopefully we will have gotten rid of any remaining residual source mass that was located on the site. Another benefit of this is it's going to give us, where we took the slabs out, some additional area for planting trees when we want to enhance the phyto system and ability to plant trees closer to where the former operations took place. So I'm going to show a couple pictures from this. This is when we were out there. This one over here is originally what the slabs looked like. You can see they're just buildings, a couple of on-site buildings. There was a bathroom, so there was even still some bathroom tile there. We dug all that up, dug up what was underneath there, brought in clean fill to fill it back up. We ended up digging it down, like I said, about 3 to 4 feet. And then here you can see a picture right here of this. This is that sump vault that they found, where it was kind of had sludgy materials around it. So we're hoping that that will help with the groundwater, downgrading groundwater concentrations as well, plus having all of that out and now restored to area that we could be using for planting. So we came back in and restored it. It was planted with grass seed and covered with straw. So hopefully we'll see some growth pretty soon out there. And our next step is going to be some phytoremediation enhancement and expansion. So I'm going to turn it back over to Amber at this point, and she's going to go through what our next step for the site is.
Excuse me. So we will be working with the South Carolina Department of Health and Environmental Control to expand and enhance the pilot phytoremediation system on site. We are likely add trees in between the plant rows to fill in the gaps, as well as put trees in under the concrete pad areas. Expanding the current system will allow for more groundwater contaminant extraction and introduce dissolved oxygen and microbes to the site. We will also look at alignment of the new trees to make sure we maximize the interception of groundwater flows and provide hydraulic control over the groundwater plume. Next slide. Enhancement of the current system will include the review of the current state of technology, such as using engineering controls and possible genetically engineered plants that have the enzymes that can break down 1,4-dioxane in their tissues. The idea of bringing the plants to the contaminants versus bringing the contaminants to the plants will be explored, as well as new regulations encouraging green technologies. We will be investigating the natural organisms in the soils at the site that are breaking down the 1,4-dioxane and determine how best to increase their numbers. We will reevaluate the use of different tree species to achieve the maximum benefits. Species such as hybrid poplars and other types of willows can grow faster and transpire large amounts of water. Eucalyptus and bald cypress are evergreen trees, and they provide year-round mechanisms versus seasonal mechanisms from deciduous trees that lose their leaves in the winter months. We will also be evaluating the installation of tree well systems, which I'll talk about in more detail in the next slide. Disadvantages for this site include a shallow water table that's 2 to 10 feet below ground surface, and the tree roots do not necessarily extend past the capillary fringe zone. Roots can't get far enough into the saturated zone to reach contaminants in deeper portions of the aquifer. The tree well systems can also be used to increase the hydraulic control in an aquifer by creating a straw effect, and I'll go into more detail about that on the next slide. So, next slide. So the tree well system is an innovative phytoremediation system developed by Dr. Edward Gatliff of Applied Natural Sciences, Inc. The system uses a specialized line planting unit with optimum planting media that promotes downward root growth, encourages contaminant treatment, and focuses groundwater extraction from a targeted depth interval. The liner extends down past the water table into the targeted groundwater zone as shown in the diagram. The groundwater is wicked up into the line's planting area to the root zone. It also allows for roots to grow deeper into the aquifer as they are seeking a water source. Once the system is installed, the soil media provides excellent growth conditions for roots and microbes. In addition, the roots of the growing trees release carbohydrates, amino acids, and other growth factors that encourage the colonization of microflora in the root zone. Aeration tubes, as shown in the diagram, are added to the tree wells to enhance dissolved oxygen levels in the soil. The soil column and rhizosphere act as a bioreactor where microbial and plant-based remediation processes, including chemical transformation, can occur. Next slide. So here's an example of a tree well system that was installed at a site in Moss Point, Mississippi, that was contaminated with 1,4-dioxane. Groundwater occurred in a deep portion of the aquifer. There were 70 trees planted in 2018 that included bald cypress and weeping willow. The tree wells were excavated to a depth of 14 to 15 feet below ground surface with a 60-inch auger. Then the bottom of the unit was sealed with 12 to 18 inches of bentonite, half on the outside of the liner and half on the inside of the liner. Bentonite was used to provide a tight seal around a piezometer that was installed inside the unit down into the aquifer, and that would eliminate groundwater from seeping into the tree well. The piezometer had a screen within the tree liner about 10 to 14 feet below ground surface, and then it had a second screen on the bottom of the piezometer deeper into the aquifer. So this, what we consider a straw piezometer, will allow deeper contaminated groundwater to flow into the tree well unit. And this basically creates an artesian condition, and there's no electricity or pump required. Next slide. So in conclusion, Parsons identified challenges at the site, including a shallow groundwater table with deeper aquifer contamination. We determined that biodegradation of 1,4-dioxane is occurring at the site. We demonstrated that phytoremediation is a viable treatment for natural attenuation of 1,4-dioxane, which is transpired through the trees and transferred to the atmosphere, where it is rapidly degraded. We removed an additional source area this year and now need to concentrate on removing the remaining mass of contamination in the aquifer. We will work with the state on a plan to expand and enhance the pilot system with new technology. And phytoremediation provides a long-term, low-cost solution for 1,4-dioxane contamination and becomes more effective as the trees continue to grow. And that concludes our presentation, and we will now take questions.
Thank you. So just to advise everyone, if you wish to ask a question on the telephone, please key *1, record your name when prompted, and press the pound key. If you wish to ask a question on the WebEx, please click on the Q&A tab, type in your question, and click Send to all panelists. So just as a reminder, *1, record your name and press the pound key, or via the Q&A tab on the WebEx. Thank you.
All right, thanks, Amber. I'm going to ask a question of my own here while we're waiting for folks to get their questions entered in. But you mentioned on the last bullet that this is a cost-effective method of remediating emerging contaminant like 1,4-dioxane. Do you have any information about comparing that cost for this method versus another standard, more traditional remediation methods?
Yes, Scott. The EPA and others have done pretty extensive analysis of cost of phytoremediation and other remediation techniques. And in most cases, phytoremediation is typically half of the traditional remedial remedies. Just Google phytoremediation and costs, and you'll get all kinds of information. For the Moss Point site that I mentioned, the engineered tree well cost for installation and O&M was about $6,500 per tree. Now, comparing that to traditional, conventional phytoremediation where you just plant a tree without any kind of an engineered system, those costs usually run about $675 per tree. So even though there's an initial upfront cost of putting in those engineered tree systems, you eventually can walk away and have a long-term solution that has minimal costs associated with after that. For the Berkeley site, Tracy mentioned to me that we've estimated that the tree well site, or the tree well installation per tree will be about $5,000 to $7,000 per tree. So if we do 10 tree wells, that would be about $70,000. Comparing that to a pump system, it's pretty cost effective.
Yeah, and I'll add to that, Scott, to some additional benefits, not just the financial, but for this particular site, it's just a big open piece of property, and we don't have any personnel working there who can do any kind of O&M maintenance for a pump and treat system. So these are completely self-running systems. We don't have to do anything. We have really shallow groundwater there, so the trees just take up water and get rained on. We don't have to go out and trim them or mow them or anything like that. So we don't have to have personnel. We do check them every twice a year or so when we're down there already to do our annual groundwater sampling. But other than that, they're really maintenance free, which is really helpful for us with a site that, is now DNR'd and there's no personnel around to go take a look at it on a regular basis. It doesn't go offline. It doesn't, power doesn't shut off, that kind of thing. So it's really helpful in that regard. And it also looks very nice because it's pretty tree stand. So as opposed to some pump and treat system or some other kind of mechanized system where you'd have equipment out there sitting, which might be, interesting to the neighbors or kids who want to climb over the fence and poke at it or take stuff and carry it off into the woods or whatever. We just have trees growing and it looks nice. The front property owner who has a warehouse on his front piece of property doesn't mind having some nice tree stands in the back, and so there's a couple other benefits too besides just the straight financial cost.
Well, a follow-up: when you have the straw in there to extract water from a deeper depth than the normal root zone, is there any rule of thumb or a limit to how deep you can get that to work?
I would say probably, I haven't seen anything more than about 30 to 40 feet. If you're going to go deeper than that, most systems that I have seen, they are usually pump the groundwater up into the tree wells, and so you have a pump that essentially irrigates the trees with the contaminated groundwater. It also depends on how fast you have groundwater recharge. You know, the piezometers. I had one site in California with heavy clay soils that to sample it, you pumped it and you came back 2 days later to be able to get enough water out of it to take a groundwater sample. So the water recharge was pretty slow. So, you know, that it depends on site conditions and the types of soils you have on how deep you can actually create that artesian effect. And I also wanted to mention some tree sites and some phytoremediation sites, if you do have deeper groundwater and you're planting either sticks or very small trees, you might have to do temporary irrigation to get them to grow for the first year or two. So some of our sites have additional costs associated with putting in a temporary irrigation system. It's just a simple drip system, but getting, you know, water to the site and then irrigating for a couple of years adds additional costs. So that also needs to be factored in, and whatever site you're going to be using.
Okay, thanks.
Mm-hmm.
Corinne, do we have questions submitted?
We have questions that have come through on the WebEx. Do you want me to read those out for you?
Yes, please.
Thank you. So your question has come through from Christine. Do you use native or non-native tree species or combo of both?
You can use a combo of both. I have several phytoremediation sites that I'm using native eucalyptus trees. They're native to the area. They're also evergreen, and they pump a tremendous amount of groundwater. So if they are native trees in the area, we will utilize them. From an engineered, you know, genetically engineered trees, which are usually the hybrid poplars, those come from usually the Pacific Northwest that are breeding these trees for their pulp industry because they're wanting fast-growing trees that they can harvest after a couple of years. And so in those cases, if you're wanting specific trees, especially if there's an enzyme in them that might break down your contamination, you would not necessarily use a native tree. But it's always ideal if you can use native trees because you know that they'll grow in that site condition and in those weather conditions.
Thank you. Your next question coming from Ted. Can you describe again how you concluded that there was a biodegradation of 1,4-dioxane?
Well, I will say that we are doing ongoing studies with that as a separate project from this, which is not our data to share. So we don't, but we do know that there are degradators in the soil that have been identified in other research studies. So we do know that there are other natural processes taking place. We also know that the native trees, as we showed, the native trees are already doing phytoremediation, so not even our installed system trees, but the surrounding trees as well. Amber, I don't know if you want to add anything to that. But...
No, that was good.
Thank you. So just to advise, there are no questions waiting at the moment. So as a reminder to our participants, please key *1 on your telephone, record your name and press the pound key, or press the Q&A tab, type in your question and click send to all panelists.
So I'll just go ahead since we don't have any questions waiting. I'll say one other thing that we had to face with the installation of this project was our regulator not being very familiar, obviously, with this technology, especially several years ago, was concerned about toxins, both like a toxic air plume from the photovolatilization out of the leaves and what would accumulate in the tree trunks if we were going to ever cut the system down. So we went through that with her. We actually did some air monitoring around and in the tree canopy, and we did not find any detections of anything. So we didn't find much mass in the leaves. I think it's because 1,4-dioxane photovolatilizes very quickly once exposed to sunlight. So there was no potential for any kind of air contamination around the tree stands. And the other thing that we decided and put in our plan was that when we do cut the trees down before we remove them, we will lay them out in the sun at the site so that any remaining mass will be photovolatilized before disposal. So that was another concern that the regulator had that we were able to address, and she signed off on it after that.
We have a couple of questions come through on the WebEx for you. Your next question is from Mark. Have exposure hazards to leaf litter woody material with contaminant in it been evaluated? Is there more impact to surface soils from fallen leaves and woody material with 1,4-dioxane in it?
So Tracy just answered that question with, concerns that the state regulator had with fallen leaf material and as well as air quality. When we did tissue samples in the leaf, concentrations were very low. So I don't anticipate that 1,4-dioxane would be any impacts with leaf litter. Woody material, typically for phytoremediation sites, if you do have organics in the plants, you cut the trees down, leave them lay on the site for a period of time, and that will cause the organics to photovolatilize.
Thank you. And then the next question is from Stephen. Have you had any experience with chlorinated organics?
So I have done, I'm trying to count up all the phytoremediation sites that I've worked on through the years. Well, interesting enough, one of my colleagues just actually sent me an article on a Superfund site with PAHs in deep groundwater. And they did a pilot study on the use of hybrid poplars, and they inoculated PAH degrading fungus into the plants before they planted them. And then they also pumped groundwater up to the trees because it was pretty deep. And so that was in hopes of creating a low-cost alternative to a costly pump and treat system. I've dealt with TCE at many of my phytoremediation sites, as well as ammonia and heavy metals. So I haven't run into any contaminants that do not benefit from phytoremediation. Hopefully that answers.
I'll add that for this site that we were talking about, we do have other chlorinated solvents, and we did see evidence of those in the trees. We just have them at such low levels at this particular site that that you don't see a ton of it taken up in the trees because there's not a direct correlation between groundwater concentrations and what you see in the trees. So we did see the other chlorinated. So this pilot is not just for the 1,4. It will also handle hopefully the other VOCs at the site. So.
Yeah, and I had a site in Utah at an Air Force base that the native trees and the phytoremediation trees had such high concentrations of TCE in them that we had to treat them as hazardous waste. And so the landscaping crew that came out to clear the brush and plant the trees things on the site. We had to get HAZWOPER trained before they could actually handle the material. And then typically when at some of the groundwater plumes, you want to plant your tree stands downstream of the hot spots in the plume. The idea being that if you get the trees planted, have a couple years of growth so they can establish their root system, that by the time the hot spot reaches the trees, then the trees are functioning and can be a part of the remediation process. So this site here in South Carolina is a little different because we are in a pretty confined space. We don't have a way to be able to remediate downstream. So hopefully with phytoremediation, we can quickly address the groundwater plume that still is on site.
Yeah, and luckily we see a pretty sharp drop-off in concentrations just based on, like we said before, natural degradation and the take-up of the native trees as well. So the existing plume doesn't extend very far off-site. You get pretty quickly dropping rates. So we're lucky in that regard that hopefully if we treat this area right on site where there's the most remaining mass, pretty quickly we should see any off-site contamination drop even more.
Are there any other questions?
We have one question that came in through the chat feature. Eric was asking, in general, what types of information are the regulators requiring for approval of phytoremediation as a closure option for a site?
Typically, I prepare a work plan that then is reviewed by the regulators. We also will do a site visit with the regulators to get them familiar with the site. We'll do a PowerPoint presentation and give them examples of where this technology is working. Because I've done a lot of work on military bases across the country, there's always air quality concerns, and that's probably one of the #1 issues I get from the state regulators of potential air quality impacts. So, having a good work plan with some examples to show how that phytoremediation system may have worked at other sites with similar contaminants and similar groundwater conditions is very helpful. And then just walking them through the process. And most regulators like the idea that it's a green technology. They're also like the idea that it has lower costs and lower operation and maintenance associated with it, and therefore, hopefully, a higher likely success rate.
Yeah, and from the management side of things, the regulators generally want to know, which is, pretty par for the course, how long is it going to be before the site is cleaned up, and what goals are you trying to reach, and how fast are you going to reach them? And so I think having strong modeling and helping to understand your site conceptual model and as much as you can, understanding how much mass you have, how many trees you're going to have, how much how much water they're going to be able to pump up. As Amber said, we can analyze that from the tree canopy. So some proposed rates of mass uptake and stuff is very helpful. It's not an exact science, and so it's difficult to say this will all be cleaned up in exactly 20 years, which is what they always want you to say. But you can come up with general rates of removal and system maintenance and how long it's going to take in an overall standpoint. And that's what the regulators were usually mostly focused on in my experience.
Okay, any other questions?
Yeah, a few more minutes if anybody has any other questions.
just to advise that there's been another one posted in chat. Forgive me if it's already been addressed. It's specifically if a tree well system is installed directly into landfill waste trench. Not all of the information, I think, is there for you, Steve, so you may want to enter a few more details for us. So Steve has just replied, thank you. Do the trees ever fall and release contaminants to the surface?
They potentially could. Where I've seen that happen, especially on heavy metal contaminated sites, where you have a plant that might be a hyperaccumulator that has accumulated high levels of metals in their tissues, and then the tree falls down and lays on the ground, and then those metals would be put back into the environment. Typically with organics, you don't see that because the analytes will volatilize or degrade once it's reached sunlight, or the soil microbes in the upper portions of the soil column might be able to degrade them. So you don't see that much with organics. Where you would see that would be if you had heavy metals, particular things that you can't get rid of.
Yeah, and you would put that into your management plan because when you're using this system, if you're using it for metals or something that accumulates, the plants are not breaking it down and degrading it; they're taking it up. So you then would have to manage the tree debris waste as waste material. So that would be part of your plan. If you're trying to remediate metals, the way you remediate them is they get taken up into the plants, and then the plants get taken away and disposed of. So it's like a removal system. Whereas for the organics, generally, the plants are actually breaking them down and degrading them. In terms of 1,4-dioxane, it photovolatilizes out of the leaves and gets broken down relatively quickly within probably 24 hours by UV radiation. And so you don't have to worry about it for the most part accumulating in the leaves and the trees because it's actually being degraded. So that would be part of your management plan depending on what constituents you had and how you dealt with that. Again, like we said before, if a tree falls on this site, we would just leave it laying out for a few days and it would be fine and we would just throw it away.
Thank you. We have a question come from Bruce. Can you inoculate the plants with 1,4-dioxane and degraders?
So if I understand the question, you're inoculating the plants with the contaminants to hopefully get the contaminants or the breakdown of the contaminants then? Is that the question?
Amber, I think he was asking if you can put the degraders in there. So if my understanding is that you're asking if you can inoculate it with the degraders. So I will say that the state of technology for biodegradation for 1,4-dioxane is in its infancy, and that's what we're working with on some other projects and stuff to talk about biodegradation and stuff. I don't think we're far enough along with that for 1,4-dioxane to be a possibility right now. I don't know, Amber, if you know for other VOCs, if you can do that. I know the fungus and stuff you did before.
Yeah, so I did a search online for this presentation just to see what the state of 1,4-dioxane technology is in terms of using either inoculants or genetically engineered plants. And so I didn't find much information on that, so I think it's relatively new field. I would imagine that it is going to be genetically engineered plants right now are a pretty hot topic, and the scientific community is pretty excited about this new technology. So I think that eventually it will happen, and you can inoculate trees with fungus, bacteria, enzymes. So if you can find something that is degrading to 1,4-dioxane, then I don't think that there is it would be prohibited to inoculate the plants with those substances.
Thank you. We have a further question come through from Adele. Any idea if this tech work will to remove metals?
Probably not. The tree well systems confine the root systems to the 36-inch or 60-inch diameter of the tree well system. In metals contamination, you want the tree roots or the plant roots, in cases of crops, to spread out as far as possible. Maybe get as deep as it can to help either phytostabilize the metals in the soils, or if you're wanting to accumulate the metals in the soils. So I'm not sure if the tree well system would work for heavy metals in soils particularly, but that would be something that you could explore. I haven't seen any cases of that, but that's what pilot tests are for, is to try out these different technologies in different situations.
Thank you. So just to advise, there are no further questions waiting at the moment. So if you still have questions, please key *1 on your telephone, record your name and press the pound key, or enter them on the Q&A tab on the WebEx and send to all panelists. Thank you.
In the meantime, can you advance one more slide? And I will let everybody know we do have one more webinar in our series for this year. We do take the month of December off to the fact that a lot of people are on holidays and things like that. But we do have one coming up for November 18th. Jeff Lormond and Natalia are going to discuss landscape architecture and urban design at Parsons and the value that that's adding to our clients. So you will see an invitation coming in a couple of weeks to that webinar, but that will be our next one, and as I said, that'll be our last one for 2020. So we hope everybody has an opportunity to join us for that. Amber and Tracy, thank you very much for the presentation. This was a great one, and we do appreciate the time and effort to put that together. We will have a recording of it that will be posted to the Parsons YouTube channel probably by the end of this week. And so we thank everybody for joining and spending the time with us. And with that, we will close out the webinar. Thanks, everyone.
Thank you, Scott, Tracy, and to our colleague Amber. So just to advise, ladies and gentlemen, that concludes your conference call for today. You may now disconnect. Thank you for joining, and have a very good day.